A very good morning. So let's move on to the next chapter. Chapter 2 is microscopy. This video is again for theory as well as for NEET PG. Now let's study about dark field microscope. Topic of chapter 2 microscopy. Principle: the object appears bright against a dark background. This is possible by special dark field condenser structure. It consists of central opaque area and a peripheral annular area. The role of the central opaque area is it prevents the light from entering the objective lens directly, and peripheral annular area which allows light to focus on specimen obliquely. Only the light reflected from the specimen enters the objective, whereas the unreflected light does not enter the objective lens. So, specimen is brightly illuminated against a dark background. Application: it is used to study the unstained living cells and thin bacteria like spirochetes, which can't be visualized by light microscopy. Let's move on to the next topic. The next topic is phase contrast microscope. In this, we have to identify the word contrast. Contrast, the contrast is enhanced, means the microscope visualizes the unstained living cells by creating difference between the refractive index of cell and water. Okay, condenser is similar to that of dark field microscope. Like we did about the field microscope, the dark field microscope, as it consists of central opaque area and hollow peripheral annular area. Okay, do you remember it? Okay, nice. Now undeviated light rays. This is a bacteria. This is a phase ring. This is your microscope condenser, and these are the eyes, the person viewing through the lens. Okay, now let's see it point by point. First, as the light rays strikes the bacteria, it gets retarded by about 1/4 due to variation in refractive index and density. Next, the phase ring is constructed. This is a phase plate. Okay, the phase plate is constructed in such a way such that the undeviated light rays, they will see this undeviated light rays will definitely strike the phase ring, whereas the deviated light rays, these deviated light rays, it will not strike the phase ring in the phase plate. Whereas this is a phase ring, this is the phase ring, the undeviated light rays strike the phase ring, whereas the deviated light rays deviate from the bacteria. It will not strike the phase ring and will pass through the plate. Third point, the undeviated light rays is definitely advanced. See this, undeviated light rays is definitely advanced by about 1/4 of the wavelength of the light. Okay, so the difference between the deviated and undeviated light rays is when it will be about half the wavelength. It will be about half the wavelength out of phase when they come out and they cancel each other to form the image. Image is formed in third point. Then we, in the fourth point, we are describing the imager. The image, the background is light, whereas the bacteria or the object appears, unstained object appears dark and well defined. Background is light by the undeviated rays. Now the applications of this phase contrast microscope, it is useful to identify the motility of the bacteria, the shape of the living cells, as well as the microbial internal cellular components such as inclusion bodies, cell membrane, nuclei, endospores. They become clearly visible. Again, the reason is very similar because of difference between the refractive index and the density between the cell internal cellular components from the water. Electron microscope. It was discovered by Ernst Ruska Electron microscope, it uses accelerated electrons as a source of illumination. The wavelength of electron is much less than visible light, so minute details of flagella, fimbriae, etc. can be seen. Okay, now it is of two types basically: transmission electron microscope and scanning electron microscope. Transmission electron microscope. For transmission electron microscope, we need to take care of three things. First, we have to deal with specimen preparation, then we have to deal with electron pathway, and then we have to deal with the applications. For specimen preparation, first go for fixation, then dehydration, then embedding, and then slicing. Fixation. The specimen is fixed with glutaraldehyde or osmium tetroxide. Dehydration. Specimen is dehydrated using organic solvents such as acetone or ethanol. Embedding. The specimen which is dehydrated is embedded in a plastic polymer and is hardened to form solid block. Slicing. The thin slices of the embedded specimen are cut using the ultra microtome knife, which are mounted on the metal slide made of copper. Now let's talk about the electron pathway. See, electrons are very lightweight, so they can collide with the air particles and scatter, due to which we use magnetic condensers. Okay. Now let's study the pathway of the electron. So first, electrons strike on the sliced specimen. Here we did the slicing of the specimen mounted on the copper slide. Okay. So electrons strike on this sliced specimen on the copper slide. Then specimen scatters the electrons passing through it. Electron beam, which is again I explained earlier that electrons collide with the air particles and scatter. So we need magnetic condensers. So electron beam, which is focused by which is passed by the magnetic condensers and is focused by the magnetic lenses to form enlarged visible image of the specimen on the fluorescent screen. Wherever the region is denser, few electrons strike, so it appears dark in the image. As the denser region, it will scatter more electrons. The applications of the electron microscope, it is used for virus detection. Virus detection of clinical specimens. For example, in the stool specimen, we can detect the rotavirus. Antiviral antibodies. Antiviral antibodies, by adding them specifically to the specimen, okay, and from tissue cultures. Then contrast is increased. For example, negative staining with heavy metals and by shadowing. Freeze etching technique disclose the shape of organelles within the microorganisms. Thank you. This is the end of the second chapter, microscopy. This video is for theory NEET PG as well as for supplementary examination of MBBS second year. Kindly like, share and subscribe. Thank you.